Okay. A very good morning to one and all. I am Rashmi Selvam, Academic Unit Representative of Bioscience and Bioengineering Department. I welcome you all to the Zooming into the World of Microbes webinar. I acknowledge all the participants for making it to the day 3 of the webinar series organized by Postgraduate Academic Council. With immense pleasure, I welcome our speaker of the day, Professor Mahuya Mukhopadhyay ma'am, Assistant Professor in the Microbiology Division of Lady Brabant College, Kolkata. Ma'am has over 14 years of academic experience and has been teaching Ma'am has over 14 years of academic experience and has been teaching industrial microbiology, biotechnology, microscopy, genetics, and other subjects to both the UGs and PGs. Ma'am has also published several papers in this domain. Today, ma'am will be delivering a talk on microscopy principles and techniques. Also, we will be uploading the webinar recording on PGAC YouTube channel. If you have any doubts, please feel free to get them clarified at the end of the session. Ma'am will be happy to answer them. Without further ado, let's begin today's session. Over to you, ma'am.
Thank you, Rashmi. A very good morning to all of you. Thanks for the warm welcome and the kind words. Let's start.
Yes, ma'am, we can start.
Yeah. Am I audible?
Yes, ma'am.
Yes, okay. So today the topic of my discussion is microscopy, the principles and applications. Okay, so I will deal mainly with the basics of different types of microscopy and their applications in the biological field. So let's start with the definition. So what we think about the definition? So it is an optical instrument. Okay, so here the involvement of light rays are there, the visualization of something is there. So it is an optical instrument and made up of either single lens or maybe a lens systems. And what is the purpose? For just making a clear and enlarged image of a minute specimen. Okay, so what is the definition? In the definition, it is an optical instrument and it is made up of lens or lens system. And what is the purpose? Just to visualize a very minute specimen. That is, we have to have a enlarged, not only the enlarged or magnified, we should have a clear magnified image of certain minute specimen, right? So now we just pass on to the what are the different types of microscope we will deal with. Okay, so this is not a classical type of classification like that. I have just classified the different types of microscope to understand it very lucidly. Okay, so on the basis of, I have already mentioned that there may be either a single lens is present or a multiple lens may be present in a microscope. So on the basis of the number of lens, we can have two different types. So either it may be simple, if only one lens is present, that is known as we call the microscope as simple microscope, and if more than one lens is present, we call it as compound microscope. Okay, and on the basis of the number of ocular lens, we can call the microscope as the monoocular microscope, and another is binocular, where more than one, that is two lenses, two ocular lenses are present, we call it as the binocular microscope. Right. So next part is very important, that is number C, on the basis of source of illumination. Okay, so on the basis of source of illumination means whether light is used as a illuminating agent or electrons are used as illuminating agent. So on the basis of that, we can call certain microscope as light microscope and certain microscope as electron microscope. And among the light microscope, there are various types. What are the types? Number one, I've already mentioned here, that is BFM, that is bright field microscope, where the field appears bright and the specimen appears dark. Clear? So this is the bright field microscope which we normally use in our daily practical works in the laboratory. So this is the bright field microscope. Next one is the dark field microscope. So from the name, it is again very much clear. Here the field is dark, whereas the specimen appears bright. Okay, so this is just reverse. So here in this case, the field appears bright and the specimen appears dark. Whereas in this case, that is dark field, the field appears dark, whereas the specimen appears illuminated, right? And number three is the fluorescence microscope, which you will obviously use in your future work. And another very important one is phase contrast microscope. Okay, so these four are the main basic types of microscope, light microscope, which we regularly use in our laboratory. Next, we pass on to the electron microscope. What are the different types? Either they may be transmission electron microscope, and another one is scanning electron microscope. Okay, so transmission. So obviously from the name, it is again to some extent clear what is the purpose, what is the mode of operation or like that. Here the electrons are transmitted, the transmitted electrons forms image in case of 10. And here the scanning, only the superficial structures can be seen with this type of microscope. So this is the overall different types of microscopes which we will discuss in our later studies. Okay, so let's start with the very basic, is the simple microscope. Then we pass on to the compound microscope or the bright field microscope. Okay, so I have already mentioned that simple microscope consisting of one lens. Okay, so it is just held in a frame and provide a stand to hold the specimen and a mirror to reflect the light. So what are the different parts? Obviously here one lens is present and which is held in a frame, and there is a stand to hold the specimen. This is the specimen where the specimen slide or the actual specimen is kept over here, and there is a mirror to reflect the light. This mirror plays a very important role in any type of microscopy. Okay, so this is a very simplified form of microscope where only one lens is involved and they are fixed to certain stand or frame, and there is a specimen holder, and obviously there is a mirror to reflect the light. And what is the application of this simple microscope? Very it is normally used in some to study the different floral parts in plants or different appendages in animals or like that. I am just saying about the application in its biological field. So it is very commonly used in the taxonomy laboratory, where the different floral parts are analyzed or like that, in case of if we study the different plant parts over there. Okay, so this is the simple microscope. Okay. So next we pass on to the compound microscope, which we are obviously you are using for several years like that, but there are something to discuss over here. So you can, if I go the slide, so you can see in this B point, on the basis of the number of ocular lens, the microscope may be monoocular or binocular. And you can see here only one ocular lens is present. Okay, so we can say that this microscope is monoocular in nature. Okay, and why it is compound? Because there are involvement of three different lens systems. Okay, so what are the different lens systems? Here, condenser lens is present, then the objective lens is present, and obviously the ocular lens. So if we are choosing certain field in the slide, you have to focus in that portion where the cells are well separated, well stained, and in the background there is no stain, right? So that is the actual ideal position of the specimen in the slide, right? So you have to choose that particular position with this 10x lens. Okay, so this is called the low power object. Then you have to focus the slide with the 40x, and finally you have to focus the slide in 100x with the 100x objective lens. Okay, so this is the different lenses with different magnification values. Next, in the ocular lens, there either maybe 10x and 15x objective lenses, maybe ocular lenses may be present. So if we want to have the magnification, we want to just calculate the magnification of a specimen you are observing, we just multiply the power of this objective lens as well as these ocular lenses, right? Suppose we are using 40x in the objective lens and 10x in the ocular lens. So we can very easily say we are observing the specimen in 400x magnification. Clear? So by this way, we can have maximally 2000 times magnification with this type of light microscope. How this 2000 times? Because in some cases, instead of 100x in the objective lens, 200x may be present. And if we are using 10x in the ocular lens, we can have this 2000 times magnified image with this type of compound microscope. And we have to say that it is the limit of the compound microscope. Why? Because if we increase the power of the lens with by using high power lenses, we can have the magnified image, but the clarity of the image will be compromised. We will get a very blurred image of the specimen. Can you get the point? So this is the magnification range or limit of the of the compound microscope, which we are using, compound light microscope, which we are using regularly in our laboratory. Okay, so just read out this portion. Condenser lens interposes between light source and the specimen and focuses light on the microscopic plane. Next, the next see, correct adjustment of the condenser lens is very important in producing a clear image by the microscope. Number 3, the condenser is positioned in order to create a parallel ray of light which illuminate the specimen. Next, objective lens produces a magnified image of the specimen, and ocular lens further magnifies the image and enables the image to be perceived by the eye. And next one, the objective lenses are a series of lenses which not only creates a magnified image, but also reduces the aberrations in shape and color of the image. So there may be certain abnormalities in the lens, and it may focus the light in various points, so we can get a blurred image if we are using the low quality lens. So there is a series of lenses are placed over in the objective so that we can minimize the aberrations created during the operation, that is, which is known as chromatic aberration or the spherical aberration, and by this way we can reduce this type of aberrations. Okay, and most of the light microscope uses 4 objective lens system mounted on a movable nose piece. 4x, 10x, 40x, and 100x are there. The objective lens bends the light which pass through the hollow body of microscope, and then one or more ocular lenses, and ocular magnifies the image created by the ocular lens another 10 to 15 times. The total magnification is estimated by multiplying the magnification of by the objective lens and the magnification by the ocular lens. Okay, so this is to some extent the introduction of this bright field microscope or the compound microscope. Okay, so in some cases the magnification ranges up to 2000x, but that is the limit of light microscope. So increasing the magnification with increased power of its lens does not produce a clear image. Resolution is restricted with the wavelength of light. We know that, and we will discuss it in the next part. The photographic image of the specimen is known as photomicrograph or micrograph, and single lens may have several optical aberrations, so a number of lenses are placed to minimize the errors. Okay, so this is the brief introduction of the bright field microscope or the compound microscope as a whole. So next we pass on to another very important part of this basics of microbiology or microscopy, we can say, is resolution. So what is resolution? It is the ability of a lens to separate or distinguish between 2 points which are placed very close. Okay, so the lens which can separate very closer points are having the higher resolution than the other which cannot separate that much closer points. Okay, suppose if we give an example, if we are dealing with a microscope which can separate 2 points which are suppose 0.2 millimeter apart, and one microscope is there which can separate only 2 millimeter apart. So obviously we can say that the first one which can separate the points which are 0.2 millimeter apart having the better resolution. And obviously the microscope is better than the latter one. Okay, so as the distance becomes smaller, the resolution of the microscope is increasing. Okay, so the term was given by Ernest Abbe in 1870s. He gave the Abbe equation for resolution, and that is the very important thing for the designing of different types of microscope, mostly the electron microscope also. Okay, so so what is the formula? D equal to 0.5 lambda by n sin theta, where lambda is the wavelength of light, n sin theta is the numerical aperture, where n is the refractive index and theta is the half the angle of light entering the objective lens after leaving the specimen. Okay, so these are the various factors which regulate the D. And I have already mentioned, if the D becomes smaller, the R will be increased. So obviously R, that is resolution of a microscope, is inversely proportional with the D. Okay, so you can see that D is directly proportional with lambda. So obviously with R it is inversely proportional. Okay, so lambda is inversely proportional with R, whereas R is directly proportional with n sin theta. Is it clear? So as the R is inversely proportional with lambda or the wavelength of light, we can say that the resolution increases with decreased wavelength of light. So if we are using lower wavelength of light, we can have better resolved image. Clear? And what about this n sin theta? Obviously, if we increase the refractive index of the media where the lens is working, we can have better resolved image. And if we increase theta, again we can have better image. Okay, so this is the basis of the resolution of any microscope. So whether we are getting clear image or blurred image, we are using this microscope or this microscope, that solely depends on all these factors. Okay, and this part I have already mentioned and I have already discussed. Next, what is written? As D becomes smaller, the R increases. So D and R are inversely proportional. Greater resolution can be obtained with shortest wavelength of light. Light, oh, this is very important. Light at blue region produces most prominent image, and R increase with with increase in numerical aperture. N is the refractive index of the medium where the lens is working. Refractive index of air is 1, whereas the refractive index of immersion oil is 1.25. Okay, so obviously if we are using the blue region of light and if we are using immersion oil instead of working in air, we can have better resolved image. Okay, because we know that just discuss in the next portion, theta is we cannot increase theta much more. Okay, so what is theta? You just see if this is the objective lens and this is the specimen, you can see the light entering the objective lens and the half the angle of this light entering is the theta. And you can see theta obviously much greater here than that part. So obviously these when we are working with this working distance, we can have higher resolution, right? So and it is again clear from this figure, the theta maximally, which is also a theoretical value, the theta maximally may be of maybe 90 degree, right? So you cannot increase theta more than 90 degree, and sin 90 degree is 1. So the only way to increase the resolution of a microscope which is working with light and can optimally produce image with blue region of light, just to increase this refractive index of the media where the lens is working. So previously we are working in the up to 40x, we are working with this air, where air is the media and having the refractive index of 1. And if we use immersion oil, which is having a refractive index to some extent similar with the glass. Glass means the cover slip or the slide which we are using, or the lens like that, so that the refractive index of the media will be increased. So we have to replace the air between the slide and the objective with immersion oil, so that the resolution will be increased. Clear? So next, just calculate this one using if we are using the 530 nanometer of blue region of light and oil immersion objective that is having 1.25 of refractive index, and we are taking theta as 90 degree, that is sin 90 degree 1. So if you calculate this, we can get this value, that is 212 nanometer or 0.2 micrometer. Okay, and this is the limit of any light microscope. Okay, so any light microscope can maximally separate 2 points which are 212 nanometer apart or 0.2 micrometer apart. So so obviously we can imagine that any bacterial cell which is having the size range of 1 micrometer or like that, they cannot be, the internal structures cannot be visible with this type of light microscope. So we have to go to another type of microscope, that is electron microscope, right? So this is all about the resolution part of the light microscope. So I think you are now having certain idea about what are the different lens system and how they are working, why we are using the immersion oil in case of 100x microscope, 100x magnification. So in case of 100x magnification, we are having a magnification range up to 1000 or 2000 times, but we are increasing the magnification, but if we does not get any better clarified or clarity in the image, the magnification does not work. So we have to create, not only in the definition I have already mentioned, in the definition I have mentioned, you have to create not only the magnified image, but you have to create the clear image also, right? So, and that part is mainly done with this immersion oil. And So in case of bright field microscope or the light microscope, what is the limitation? We can get maximally 2000 times magnification, as well as we can separate the 2 points which are 212 nanometer or 0.2 micrometer apart. Okay, so up to this, I have just done with the bright field or the compound microscope. Okay, so next pass on to another type of microscope. Do you have any question in this part, or you just discuss the questions after the session? Please unmute and tell me. Yes, yes.
Good morning. So there was a question from Prasoon. So Prasoon, if you would like to ask the question,
Yeah.
please.
Hi, ma'am. Hariom, ma'am.
Hi.
Actually, I guess in the dark field microscopy, the question will be answered, I guess. So,
okay. So I should proceed, right?
Yeah,
okay.
Sure, ma'am.
Okay, thank you. Okay. So next we pass on to the dark field microscope. So I am presenting in very lucid way. Okay. So that you can just having a overall idea of that type of different types of microscope. Okay, so if we see on the on this part the light pathway, right? So this is a light pathway in the bright field microscope, and this is the light pathway in the dark field microscope. So in the bright field microscope, you can see if this is the light source, you just recollect the picture I have already shown you with the bright field microscope, whether it is binocular or it is monocular. There is at the base there is a light source and condenser lens, then the specimen, then the objective lens, then the ocular lens, and finally the image is formed in the eye, right? Or you may photograph it with your device. So this is the light pathway of the bright field microscope. Okay, so in case of dark field microscope, there is some alteration. So what is the alteration? So in between the light source and the condenser lens, there is a opaque disc is present. Okay, so what is the function of this opaque disc? It just blocks some light rays and it creates a hollow cone of light, right? So this is the difference in between this bright field as well as dark field microscope. So here an opaque disc is present and the light is focused in the specimen in such a way that the unreflected and unrefracted light does not enter the objective lens. Am I clear? So the light is focused in such a way that unreflected and unrefracted light does not enter the objective lens, and the light rays which are reflected or refracted by the specimen only enters the objective lens. Am I clear? So here the light is, the specimen is illuminated in such a way, okay, so that the unreflected and unrefracted light does not enter the objective lens. Only the light rays which are reflected or refracted by the specimen enters the objective lens and ultimately forms the image in the eye. So obviously the specimen appears illuminated, whereas as the unreflected light does not enter the objective, the field appears dark. Clear? So this is the basic difference between the principles of this bright field as well as dark field microscopy. Okay, so here the specimen appears illuminated, whereas the background becomes dark. Unstained. So this is very, very important because in your staining class you have read about different methods of staining. But why such staining is required? Staining is mainly required just to create the change in the I can say in the refractive index, right? So just to create certain contrast between the specimen and the background, right? So in the dark field microscope, what happened? You just see the bright background and stained specimen. But here you does not require any staining technique, because here the mode of operation of this microscopy is totally in different way. So unstained specimen can be seen with this type of microscope. A hollow cone of light is focused on the specimen in such a way that unreflected and unrefracted light do not enter the objective. Only the light rays that have been reflected or refracted by the specimen forms the image. The field surrounding the specimen appears dark, while the object itself is brightly illuminated. So this is the basic point of the dark field microscope. Okay, so what are the applications used for examining the living cells? The microorganism that cannot be stained by the standard methods or are so distorted by normal staining methods so that the characteristics cannot be identified. So in that cases, this dark field microscope is applied. Very thin spirochetes like Treponema pallidum can be identified by this type of stain, sorry, not the staining method, by this type of microscopy, I can say. Okay, and this is the pictorial presentation. So how it looks under the dark field microscopy. I think when you are doing with the staining class, the negative staining one, right? So in that case also the background appears like that and the specimen like that. But the main thing is that here you can see the live cell also. Okay, so I think I give some answers to your query regarding dark field microscopy, right? So I have completed the two different types of microscope, that is bright field as well as dark field. So should I proceed to the next one?
Ma'am, can you just show us the last slide, the applications of the dark field microscopy?
Yeah.
Just wanted to take them down. Just give me a minute.
Okay.
All right. Thank you, ma'am.
Okay. So today I will discuss about up to this fluorescence microscopy principles, rather the construction of fluorescence microscopy and the application part of fluorescence, as well as the phase contrast and other two electron microscope I will discuss tomorrow. Okay. So I have just designed in that way. Okay. So
ma'am, sorry, ma'am, sorry for interrupting. Can you just show me the slide for a minute? Just wanted to take them. Yes, ma'am. I just wanted to take down the points.
Oh, okay.
Thank you, ma'am.
I will share the content with you, right?
Okay.
For all the
All right.
participants, this video will be available on the official YouTube channel of PGAC. In case you miss out on any part, you will get to all these things.
This video, as well as I will just send this presentation to Shrohini also. Okay, and Shrohini, please share it with your friends.
Yes, sure, ma'am.
Okay.
All right. Thank you
So let's proceed. Okay. So next part is very important one, is the fluorescence microscope. Okay, so I have to first discuss the different parts. What are the function of that of the different parts are like that. Okay, so and the application part I will discuss in the next class. That will be better. Or if I get time, then I can also. So it is used to study the properties of organic substances using the phenomenon of fluorescence. Okay, this is designed in such a way that the specimen can be illuminated with light of a wavelength which causes fluorescence in a sample. The light emitted by the dye, which is at a different longer wavelength than the illumination, is then detected through the microscope object. Okay, so there are some something different than the other types of microscope. Just simplified way, you can say that I can say that here certain dyes, which are known as fluorescent dyes or fluorochromes, are normally used. Okay, and these fluorochromes having affinity with certain parts of the cells. Some dyes may bind with certain specific types of microorganisms. Some may tie up with different structures within the cell, like that. Okay, and what is the this is the property of the fluorescent dye. And another very important property is that it just absorbs the light at a specific wavelength and it emits light in a certain higher wavelength. Okay, so there are two different light sources. The wavelength of light are two different. We are using the incident light, which we are saying that as excitation light, and we are getting in certain wavelength, and we are getting certain light waves which are certain than the higher than the excitation wave. Am I clear? So just see this picture. This is the fluorescent microscope, and you can see it is to some extent obviously some complicated one, and this is the ray diagram of this fluorescent microscope, I can say. Okay, so just discuss this picture, and you can observe, and you can I am sure that you can understand the mechanism of this fluorescence microscope. Okay, so first of all, suppose this is a light source. So as it is a light source, it is producing a whole spectrum of the light, right? So, and I have already mentioned this fluorescent dyes. So the specimen is first stained with the fluorescent dye. Okay, and this specimen and this dye having the affinity with some particular wavelength of light, and that is the excitation wavelength. Suppose we are using here the blue region of light. Obviously, blue region of light is much lower in the visible range. So we are using, normally we use this blue region or the UV region of light with this to excite the specimen. Okay, so here blue region of light is required for excitation. And here you can see there are presence of two filters. One is excitation filter and another is emission filter. And another very important thing is there is a presence of dichroic mirror. Okay, so this is the excitation filter, right? So what is the function of this excitation filter? The function of this filter is that there is a combination of different wavelength, and it just filters the required wavelength, right? So it just allowing the blue light to come through this object and fall on this specimen. Am I right? So after absorbing this blue light, the specimen, which is already stained with some dyes, will emit certain lights, which is known as emission wavelength. So excitation wavelength and another is emission wavelength. What I have mentioned, that is excitation wavelength is much shorter, whereas the emission wavelength is much higher. Clear? So now this emission wavelength will pass through this emission filter, and ultimately it is detected through our eyes or the photographic instruments or like that. Okay, so how it is? So the function of this emission filter is that it just stops the excitation light and only allows the emission wavelength, and the image is formed in the detector. So what is the function of this dichroic mirror? See, from the name here, the terminology is dichroic, means to color. Okay, so a very important role is played by this dichroic mirror. Each and every dichroic mirror having a transition wavelength. Okay, so what is transition wavelength? It is 50%. Suppose we are using this light at 500 nanometer, and it is emitting certain light which is in 600 nanometer. Okay, so this dichroic mirror having a transition wavelength value at 550 nanometer. Okay, so the function of this dichroic mirror is that it will reflect the light which are below the transition wavelength. Am I clear? And it just transmits the light rays which are above the transition wavelength. Okay, so as this excitation wavelength having the, just for example, having the wavelength value of 500 nanometer, which is less than 550, which is the transition value of this dichroic mirror, will be reflected and will pass through this direction and fall on this specimen. Whereas the light which is emitted after excitation, is the emission filter, is the emission wavelength, and having suppose 600 nanometer wavelength, and which is much higher than the transition value of this dichroic mirror, and will pass to the dichroic mirror, transmits to the dichroic mirror, and ultimately through the excitation filter, and ultimately reaches the detector, right? So, but now the question arises, then why we are using this emission filter? Because dichroic mirrors totally stop the passing of this excitation light to the detector. So why we are using this emission filter? The answer is that dichroic mirror is only 90% efficient. Okay, so what is 90% efficient? Because there are 10% probability, okay, of transmitting the light which are of lower than the transition value, and reflecting the light which are above the transition value. So this dichroic mirror is to some extent 90% efficient to separate the two light pathways. That is the excitation and the emission. So these three special equipments are doing their task very carefully, and we can have a very good image of the specimen. Okay, so am I clear? You can see again here from this specimen, this construction, mainly the light source is present over here. Okay, and there is a filter over here, placed over here. And dichroic mirror is present in this location, so that the light rays which are coming from this side will be reflected to the objective, and here the specimen is present, which is already stained with some fluorescent dye, and it will excite the specimen, and ultimately after excitation it will emit some light waves which are of higher wavelength, and it will pass through the dichroic mirror. Here the emission filter is placed, and ultimately it is viewed by the eye, by our eyes, or it may be photographed through this part. Okay, am I clear?
Excuse me, ma'am.
Yeah.
Yeah, so why do we need an emission filter? In the sense that it only, filters out light of certain wavelength. So won't that decrease my intensity of light from the sample?
No, not for decreasing. For the emission filter is required, because I have already mentioned that the dichroic mirror is 90% efficient. So there is probability of 10% excitation light may enters through this may transmitted through this dichroic mirror, and may interfere with the image formation. Am I clear? See, this is 90% efficient, so there is a 10% chance of this blue light may pass through this dichroic mirror and may interfere with this green light. Obviously, two light waves, if the two different light wavelength of light is coming to our eyes or in the detector, it may interfere with each other. The clarity of the picture may be hampered. Okay, so these 10% light, 10% incident light or the excitation light is filtered through this emission filter. Okay, so only we get the emission wavelength and a very clear image of the specimen we can have. Okay, so this is the this is mainly the principle behind the fluorescence microscope. So within this microscope, it can separate the two light pathways. Okay, one is excitation, which is of lower wavelength, and another is emission, which is of higher wavelength. Another thing I have to mention, in some cases, UV light is also used for exciting the specimen, exciting the dye. Okay, and if it is excited with the UV region of light, it will emit the light at a visible region. higher, which is having the higher wavelength values. And if due to that 90% efficiency, this 10% UV light may come to our eyes, which may be detrimental. So that this emission filter is very much required. Okay, so this is the basic principle of this fluorescence microscopy. Okay, there are two filters, excitation filter and emission filter. So excitation filter which ensures the illumination is near monochromatic and at a correct wavelength. It is placed just prior to the dichroic mirror. It omits the other wavelength and blocks them and only allows the excitation wavelength. Okay, that I have already mentioned, because this is a monochromatic light, whereas when the light is produced with this with this source, it is polychromatic. So it just allows the required light. Okay, so that is the function of this emission filter, and only allows the excitation wavelength. Emission filter, which ensures none of the excitation light reaches the detector. It is placed to more specifically select the emission filter and removes traces of any excitation light. It is just placed above the dichroic mirror. And dichroic mirror, it is a special reflective, special reflective properties allows it to separate the two light paths. Each mirror has set of wavelength value called transition wavelength of 50% transmission. The mirror reflects the wavelength of light below the transition wavelength and transmit the wavelength above it. But it is 90% efficient. Okay, so these are the various, these are the basic principle of the fluorescence microscope. So today I will discuss up to this, and or should I continue? Is it 12 o'clock?
It's almost
Okay.
12 o'clock now.
Okay, so should I continue or stop here?
Ma'am, anything that is convenient for you.
Okay. So just do that. So fluorescent dye, what is fluorescent dye? A fluorophore is a fluorescent chemical compound that can re-emit light upon light excitation. Fluorophores typically contain several combined aromatic groups or different types of cyclic compound molecules. Many fluorescent stains have been designed for a range of biological molecules. Some of these are small molecules that are intrinsically fluorescent and bind a biological molecule of interest. Major examples of these are nucleic acid stains like DAPI, waste, and phalloidin, which are used to stain the actin fibers in mammalian cells, and other includes like acridine orange, acridine yellow, auramine O, Nile blue, Nile red, etc. Okay, so these are the various different types of fluorescent dyes or fluorophore which are available and normally used in the biological field. Okay. And there are obviously a huge list of applications. So the DAPI binds with the minor groove of DNA and does label the nuclei in the cell. Nile blue and Nile red specifically bind with the polyhydroxyalkanoid molecule in the cell, thus can be easily detected under microscopy, which is a very specific. I will discuss it a little bit in tomorrow's class. Mycobacterium tuberculosis can be detected by auramine O, Bacillus anthracis can be identified by this dye, and there are also another method which is known as fluoro antibody technique, which I'll discuss in the next class also. So you can see, I'm just showing these two figure and conclude. And this is the slide which shows some stains which are specifically bind with this fluorescent dye. Okay, and you can see very fluorescent green color of the specimen, and it just observed under the field. And this is again another slide which showing the staining of these specimens with this type of fluorescent dyes. Okay, so today up to this, and fluoro antibody technique and other different types of applications I will discuss in the next class. Okay, so if you are having any questions regarding up to this, you can ask.
Ma'am, I had one question.
Okay.
Ma'am, I have one question.
Yeah, please.
Usually, whenever we see a specimen that is a bit thicker, we use confocal microscopy in place of the fluorescence microscopy, and so that we take the specimen scanning in batches so that the background noise can be decreased. But when we, I just do not know what about the microbes. We usually take that for plant specimens. So when we consider any Mycobacterium tuberculosis, as you said, using the fluorescein dye and all, in that particular case, is it that we prefer confocal over fluorescence, or I do not know how do we think in terms of a microbe? Like, what is the thickness and in terms of microbe?
Yeah, as the thickness of the microbes are much smaller or much little than that of the plant tissue, it's preferable to see them very easily in fluorescence microscope. Okay, and for plant tissue, obviously the thickness is much higher. So we normally we go for the confocal as the thickness varies or like that. But in case of microbes, it is very easily seen with the fluorescent microscope.
Okay. All right, thank you so much, ma'am.
And this is Sweta Rani. What determines the transition wavelength of the dichroic mirror? Does it remain constant? If yes, how does it work for the specimen having different emission wavelength? Yeah, the specimen with different emission wavelength. Not only it is not very much specific, as I have mentioned, it is 550, not like that. It is having a range of transition wavelength. Obviously, the dye we are using having the excitation wavelength is much lower than that of transition, and we should know the emission wavelength of that dyes also. Okay, so obviously it is not a very specific one, it is a range one. Okay, anything else?
In case,
yes.
So yeah, my question is, when we have to observe a sample under the microscope, what are the conditions to consider? When should we go for light microscopy or for dark field microscopy?
Yeah, if you are using any sample which are dead, which you are applying stains, okay, then you proceed for the bright field microscope. And if you have to observe certain microscope which are very delicate, and you have read something about the microscope, that is its shape is suppose rod shaped, but if you are staining with certain techniques, you are seeing under microscope, when you are observing it under microscope, it becomes suppose distorted shape. So you have to conclude that the staining technique is not ideal to observe that microscope. So if you are if you just wish to observe them in its native state, in its living state, you have to use the dark field microscope. Otherwise, for the normal work with the culture, you are applying stains or like that, you just use the bright field microscope. Okay, microscopes, and we just learn it in great detail about the bright field microscope, mainly what are the parts, what are the function of the different lenses, like that. And we deal with the magnification and the resolution part, and then we pass on to the dark field microscopy, and finally we are with the fluorescent microscopy. And we have learned in great detail about what is the function of the excitation filter, dichroic mirror, and also the emission filter. But there was a little doubt about what are the number of filters which are used over there. So I have something to share with you. So again, go with this fluorescence microscopy, and here you can see in the fluorescence filters, there is a filter block tar present. Okay, so here in the each block, this is the we can say that optical block or filter cube. Here the not only the excitation filter is located, along with each and every excitation filter, there is a dichroic mirror and obviously a barrier or emission filter. So when we are changing the excitation filter, we are changing the whole block, where the dichroic mirror as well as the barrier filter is also changed. And you can see over here different blocks are present in the filter block tart. So when we are just changing each and every block, so we are changing both the dichroic mirror as well as the emission filter over there. And I have just accumulated data from a site where they are just selling the different types of filters, and from that data you can also see if we are selecting blue fluorescent protein. Okay, we are selecting some excitation filter for blue fluorescent protein which having the wavelength of 390 nanometer. Okay, and in that case the emission filter you get at 460 nanometer. Okay, can you get the point? Here you can see the excitation filter with blue fluorescent protein, you are getting 390 nanometer for excitation, and for emission they are suggesting 460 nanometer. And again, if we look at the dichroic mirror, this blue for this blue fluorescent protein, you can see that the reflection band is in between 360 and 407, whereas the transmission band lies in between 425 and 475. Is it clear now? So see, here the emission filter, just emission wavelength is 460. So here the dichroic mirror in the transmission band is 425 to 575. Clear? So for each and every type of excitation filter, there is a separate dichroic mirror along with the separate emission filter, and you have, when you are just changing the tart in the fluorescent microscope, you are changing the whole setup. Okay, this is one type of arrangement. And as you suggested in the previous class, one of the participants, you were suggesting that there are lots of videos are available. So I just gone through some of the videos, and here I found some authentic one where where there is a discussion about that type of cube. And again, I found something that is where multi-band pass dichroic mirrors are used. So in that case, if we are just select any of the excitation filter in a tart or in a wheel like that, we can use a single dichroic mirror which can selectively reflect or selectively emit a particular band of wavelength. And not only the particular band of wavelength, it has several band of excitation and emission to select several bands of excitation as well as emission filters. So in that case, they engineered certain filters which are having multi-band pass capacity. Okay, so this is known as multi-band pass dichroic mirror, and in that case also you can use either multi-band pass emission filter or second filter wheel. Okay, in case of emission. So we are using excitation filter wheel over here, where different types of filters are present to select a particular excitation wavelength, and you may use a multi-band pass dichroic mirror, and you may use the multi-band pass emission filter or second filter wheel also. Okay, so I think there may be in some advanced type of fluorescence microscope which are recently available, there may be such type of arrangement over there, but normally conventional microscopes are with such type of block, where when we are changing the tart, we are changing the cube, we just changing the whole set of excitation as well as the dichroic as well as the emission filter. Okay, so such type of arrangement are normally encountered in the fluorescent microscope. So next we pass on to the application part. Whatever I have written here, I have already mentioned to you, and I have to discuss the direct fluorescent antibody technique and the indirect fluorescent antibody technique. I have just briefly described it. And what is direct fluorescent? Here we use the fluorescently labeled antibody to bind and illuminate a target antigen. Okay, so the antibody itself is bind with a dye, and it will be bind with certain antigen. so that we can, when we observe the slide under microscope, we can detect the antigen. So direct fluorescence antibody tests are particularly useful for rapid diagnosis of bacterial diseases. For example, fluorescent level antibodies against Streptococcus pyogenes can be used to obtain a diagnosis of strep throat from a throat swab. The diagnosis is ready in a matter of minutes, and patient can be started and on antibiotics before even leaving the clinic. So when we are taking this technique as a detection tool, that we can easily, that is immediately, we can identify whether that type of antigens are present or not, and we can advise the patient for immediate administration of the drug. So it is very much effective in diagnosis of certain diseases. Okay, so DFA techniques may also be used in diagnose the pneumonia caused by Mycoplasma pneumonia or the Legionella pneumonophila from sputum samples. The fluorescent antibodies bind to a bacteria on a microscope slide, allowing ready detection of the bacteria using fluorescence microscopy. Thus, DFA technique is valuable for visualizing certain bacteria that are difficult to isolate or culture from the patient sample. So it is a very easy and very ready diagnosis of certain antigens or bacteria in a patient sample. So it is very efficient technique one. And another is the indirect fluorescent antibody method. Tests are used to look for antibodies in patient serum. Not the antigen we are searching for the antibodies in the patient serum. For example, an IFA test for the diagnosis of syphilis uses Treponema pallidum cells isolated from a lab animal and a smear prepared on a glass slide. Okay, so initially the syphilis, which is an antigen, is prepared in a glass slide, and a smear is prepared, and patient serum is sprayed over the smear, and anti-treponemal antibodies, if present, are allowed to bind. So if in the slide syphilis is present and in the patient blood anti-treponemal antibodies are present, then they will bind with each other. Okay, the serum is washed off. So after binding, after allowing for some time, the serum is washed off and a secondary antibody added. The secondary antibody is an anti-human immunoglobulin conjugated to a fluorogen. On examination, the Treponema pallidum bacteria will only be visible if they have been bound by the antibodies from the patient serum. Okay, so this is the indirect method. Here, not directly we are tagging the antibody which are reacting with the antigen, but we are tagging, we are concerning about that polyclonal antibodies, and they just tagged, and ultimately they are allowed to react with the antibody which already bind with the antigen. So it is an indirect method. So you can see is a pictorial presentation of such type of IFA technique. Here antigen is fixed to a slide, to a surface, then patient serum is added, and if antibodies are present, they bind to the antigen, and secondary antibody with fluorescent label is added. If patient antibodies are present, the secondary antibody binds to the patient antibody. So it can also very easily detected under the fluorescent microscope. So these are some of the applications of fluorescence microscopy. So we can either use the microscope for different types of for identification of different types of microorganism. Not only that, it can bind specifically with different parts of the cell, either with the DNA or with the other part like that. And we have also mentioned about the different types of reserve materials like the polyhydroxyalkanoids. So what is polyhydroxyalkanoid? It is one type of carbonaceous reserve material, and they are having if they are extracted and tested, and we can find that they are having some properties which are very similar with the polypropylene. Okay. Also, there was a trend in some years back that if these polyhydroxyalkanoids are isolated from the bacterial cell and maybe used as a substitute for the present day plastics. So, so it was a very trend for some years research, obviously, but the mechanism was very expensive, so it cannot be used as a substitute for plastic even. But that particular polyhydroxyalkanoid molecules, granules, can be easily detected by staining with this Nile blue or Nile red stains, and can be and we can identify the organisms which are high producer of this type of particular molecules. Okay, so such type of measurement or such type of visual measurement, I can say, can be done with these fluorescence microscopes. And not only that, it has some clinical applications also, which we have already mentioned. So this is overall about the fluorescence microscope. So what are the main things were there? Mainly there were certain dyes, okay, which having some capability to absorb certain wavelength and have to emit certain higher wavelength of light. And that particular feature is exploited in the fluorescence microscope, right? So next we pass on to another type of very important light microscope. Oh, again, another thing I have to mention, the confocal microscope is to some extent a type of fluorescence microscope where a pinhole is used, right? So what is the function of the pinhole? In fluorescence microscope, we get fluorescence from different levels of the samples. Okay, so if is a thick sample or whatever the sample, the light is coming from different planes, but which may interfere with each other, and the clarity may be not up to the mark. Okay, and that may be replaced by the confocal microscope, where we are using certain pinhole that is before the excitation filter as well as after the emission filter. Okay, so that we can view a particular point at a particular time. Okay, so there is no interference from the surrounding light waves, so that the image is crystal clear. Okay, so that is and in that confocal microscope also we are using certain dyes, and there is a presence of excitation filter, dichroic mirror, the emission filters, all are there. And along with that, what is the extra feature? There is a presence of pinhole, so that only a particular point is illuminated at a time, and we can have a clear view of that particular point, and there is no interference from the surrounding lights. Okay, so it is much better than the fluorescence microscopy. Obviously it is expensive than the fluorescence microscopy, so whatever you are available, you have to do with that. Okay, so next this is all about the fluorescence part. Next we pass on to the phase contrast part. Okay, so from the name, I think it is very much clear that is what is the function of this microscope. Okay, so it has certain features. This microscope having certain features which allows the microscope to create a contrast in phase of light waves, right? So what are the special features which are present in the phase contrast microscope? There is a annular diaphragm or annular stop or opaque disc, whatever you are saying, in the just below the condenser. Okay, so it creates a hollow cone of light, and so this is present in between the light source as well as the condenser lens. So a annular diaphragm is present, and after that there is a specimen chamber or specimen holder where you can place the specimen in a slide. Then there is an objective lens, and obviously there is an ocular lens, and in between this objective lens and the ocular lens, a special phase plate is present. Okay, and the presence of this phase plate is mainly or solely responsible for creating a contrast in the light waves which are passing through this specimen and ultimately the phase plate. So how the phase plate looks? It looks like this. Okay, so it is having such structure, that is, this portion and the center portion are coated with certain light retarding material. Clear? And this is the only ring where the portion devoid of light retarding material. Clear? So the light waves which are passing through these and this portion are retarded, and the light which are passing through this portion are not retarded. Okay, so this by using such phase plate in that portion, we can change the phase of light. So some light rays may be out of phase, some light rays totally out of phase, some light rays may be slightly out of phase, and by this way we can have a change in the phase of light which creates the contrast. Okay, so there is no need to stain the specimen, but by just placing this phase plate or the phase ring, by the way the light passes through this phase plate or phase ring, we can create a contrast. Okay, and that is the principle of this phase contrast microscope. Okay, so how such phase difference is created? You can see there is a lamp. Okay, obviously is light source, and after that and the annulus or the diaphragm is present. And obviously it is you can recall from the dark field microscope, it just creates a hollow cone of light, right? Then the condenser is present, and after the condenser there are specimen plate. And here you just think of it, if in the specimen plate there are certain, suppose we are concerning about certain bacteria, so some light rays may encounter these bacterial cells, and some light rays which does not encounter the bacterial cell. Okay, so the light rays which are which encountered the bacterial cell, we can say it as diffracted waves or deviated waves, right? And the light rays which are not encounter the bacterial cell, we can call it as undeviated rays or the surrounding rays, clear? Or surrounding waves, we can say. And here you can see that blue light, blue marking are here. That is the deviated one. Okay, and that deviated light rays, they are passing through this portion of the phase plate, and they missing the phase ring over here. Okay, so phase ring, which portion is the phase ring? This is the phase ring, right? And these deviated rays are passing through this portion. Okay, so obviously the deviated rays initially they are retarded by the wave by the refractive index of the bacterial cells, and again they are retarded by this portion of this phase ring. Okay, and these rays which we call as surrounding rays, okay, they are not retarded or not deviated by the bacteria, and they are passing, if they are passing through this phase plate in the phase ring, what happened? They are not retarded because that portion is not coated with certain materials which retard the light waves. So what happened? These light rays, these incident light rays or deviated light rays are initially retarded and again retarded, whereas these light rays are not deviated or retarded first, and again they are not retarded or deviated first. So obviously they will reach here earlier than that of the this deviated light. Okay, as they are retarded here and also there. Clear? So here the light rays first, when it was started, they were in phase. Okay, and after they reaching here, they are out of phase. Whether they may be fully out of phase, there is a phase difference of half, or they may be retarded by 1/4 wavelength, or they may be retarded to some we can say slightly out of phase. So there is certain phase difference created in these light waves after passing through the specimen, as well as after passing through this phase ring in the phase plate, or phase plate in the phase ring, we can say. Okay, so this change in the phase of light creates a contrast in the image, and we can visualize the bacterial cell or whatever the things we can visualize with high contrast. Clear? So this is the basic principle of this phase contrast microscope. Okay, here also you can see, you can very clearly observe that the light rays are in phase initially. So after passing, they are retarded, and again they miss this portion in the phase plate, in the phase ring portion, and they are, as these portions are mainly this portion is covered with some light retarding material, they are again retarded. So there is a clear phase difference over here, and they ultimately forms the image. Okay, so by this way the phase difference is created in phase contrast microscope, and that is the phase contrast microscope is creating a contrast in the slide or in the specimen. So by this way, the undeviated rays are encounter a phase ring in the phase plate, special optical disc located in the objective, while the deviated rays means the ring and pass through the rest of the plate. The deviated rays are again retarded, so they come together to form an image. So this is the basis or the principle of the phase contrast microscope. And what are the applications? It is useful in studying the microbial motility. The shape of the living cell can easily be viewed with this type of microscope, detecting various different bacterial components like endospores, inclusion bodies, polymetaphosphates, etc., and used to observe the cells cultured in vitro during mitosis. And it is also used in growth dynamics and behavior in a wide variety of living cells in culture. So this is all about the phase contrast microscope. So I am just touching only the basics and the principles of this type of microscope. So the principles as well as the application is done. Okay, so next we pass on to the electron microscope. Oh, these are some images which are clicked through the phase contrast microscope. Okay, so we have completed with all 4 different main types of light microscopes, that is bright field, dark field, as well as fluorescence and phase contrast. So next we pass on to the another very important one, that is the electron microscope. So just the first part, the introductory part, the human eye having the resolving power of 0.2 millimeter, whereas bright field can differentiate which are the objects which are 200 to 250 nanometer apart, whereas the electron beams having wavelength of 0.005 nanometer. Okay, so in you can recall that when we are calculating this 200 to 250 nanometer resolution, we are using the blue region of light, that is approximately 500 nanometer. So electron beams having the wavelength of 0.005 nanometer. So which is nearly 1 lakh times shorter than the light waves. Okay, and we are applying here again the Abbe equation of resolution, that is D equal to 0.5 lambda by m sin theta. So whenever we are decreasing the wavelength of light or decreasing the wavelength, we can get the better resolved image. So we are using certain beams which are 1 lakh times shorter wavelength than that of the light waves. So obviously we should have better resolved image. That is the main point or main theory of the electron microscope. So having a resolving power 1000 times greater than the light microscope. it can separate two points which are 5 angstrom or 0.5 nanometer. Okay, so this is the very important thing regarding the electron microscope construction. So I am just giving you the brief detail, or not the detail, brief idea, and I will discuss on mainly on the basis of differences of this electron microscope with the light microscope, not going in much detail. Okay, so what is the if we just placing the images or light pathways, light microscope, the transmission electron microscope, and the scanning electron microscope. So these are the construction of these three different types of microscope. We can view that in this light microscope we are having the light source, whereas in case of transmission as well as the scanning electron microscope, we are having certain source of electrons, mainly we call it as the electron gun, which is made up of tungsten filament mostly. Okay, so here again the condenser lens is present, and you can see in the transmission electron microscope here also the presence of the condenser lens. And the main difference is that here we are using the glass material, whereas in case of electron microscope we are using the magnetic lenses, right? And here the specimen is normally we place it in a glass slide, but here we are using certain some specific type of copper grids. Okay, I will show it in the later in the later part. And in the objective lens is again present, and here also the objective lens is present, but obviously the objective lens magnet is present. And here you can see the image in the eyepiece, the ocular, and here the projection lens is present, and ultimately we can view the image in the fluorescent screen. And this is if we consider the construction of light microscope and the electron microscope, they are more or less the construction wise similar. Okay, but in case of scanning electron microscope, the construction is to some extent different. And I have already mentioned that in the transmission electron microscope, the primary electron which are produced during the from the electron gun, they are passing through all the lenses and ultimately passing to the specimen and ultimately forms the image. But whereas the main difference from the TEM to SAME is that the primary electron passing through the different lens system, they just hit over the surface of the specimen, and from that surface again the secondary shower of electron are generated, and that secondary electrons are then captured by the detector, and ultimately the image is formed. So we can have a inner view of the specimen with this transmission electron microscope, whereas we can have the surface nature, the three-dimensional structure we can have in this scanning electron microscope. So this is just a very basic and brief idea about the light microscope and what are the about the electron microscope and how they differ from the normal operating light microscope. So the transmission electron microscope operates on many of the same optical principles as the light microscope. The TEM has the added advantage of greater resolution. This increased resolution allows us to study ultrastructure of organelles, viruses, and macromolecules. So obviously we can have, as we are having a greater resolution, we can have the much detailed structure of different organelles. Specially prepared materials samples also be viewed in the TEM. Since electrons are very small and easily deflected by hydrocarbons or gas molecules, it is necessary to use the electron beam in a vacuum environment. So the operation is totally taking place within a vacuum environment, and there are several types of pumps are active over there. So a series of pumps are used to accomplish the adequate vacuum for this purpose. The illumination source or the electron gun is a thermionic emission. In a thermionic emission TEM works such like a light bulb. A filament is the source of electron. It is usually hairpin shaped tungsten wire. an accelerating voltage is applied to the surrounding cathode cap. A small emission current is then applied to the filament to achieve the release of electrons. The point at which the gun achieves good thermal emission as well as an acceptable filament life. Okay, that is called the saturation. We are applying certain current, but it is giving very high amount of emission, but the life of the filament is reduced. That is not acceptable. We should have certain acceptable lifespan of the filament, as well as we should have the emission. Okay, we should have the optimal emission of the electrons, and that particular voltage is taken, and that is the saturation point of the excitation. The electron microscope lenses are electromagnetic converging lenses. Intermediate lenses magnify the image coming from the objective lens. Finally, projector lenses further magnify the image coming from the intermediate lens and projects it on the phosphorescent screen. Okay, so this is about the brief introduction, and here I am just showing you the instrument, the microscope over here, and this is the cross section of the microscope, and you can have the image like this. Okay, so next, what is the application? Transmission electron microscope is ideal for a number of different fields such as life sciences, nanotechnology, medical, biology, and material science, forensic analysis, gemology, and metallurgy. The TEM provide topographical, morphological, compositional, and crystalline information. The images allows researchers to view samples in a molecular level and making it possible to analyze structure and texture. So these are the various applications of TEM. And if we pass on to the scanning electron microscope, it is used to examine the surface structure. That produces images from electrons emitted by the object surface rather than from the transmitted electrons, which I've already mentioned. When the beam strikes a particular area, surface atoms discharge a tiny shower of electrons called secondary electrons, and they are trapped by a special detector and forms the image. Okay, so this is about the same, and obviously the same renders a detailed 3D images. Extremely small microorganisms can be seen. The anatomical structure of the insects, worms, spores, and other organic structures can be visible with the same. So these are and this is also the some other applications of same. So not only by this way we can use different techniques like the shadowing, pre-fracturing to have certain surface structure and 3D model of tiny elements, and we can analyze them with the electron microscope. Okay, so last part, and this is the instrument, and this is the picture we get after observing under same. So the last part of my discussion is what is the difference between the specimen preparation technique in our light microscope as well as in our in electron microscope. We have already in your previous lectures you've gone through the specimen preparation technique in the light microscope. So what were the steps for that preparation? First, you have to take a slide which is grease-free in nature. Then you have to prepare a smear, then air dry, then heat fixation, then application of certain stains, and then washing, finally air dry and observing, right? So these are the normal methods which are used for any type of preparation in the light microscope. And if we are using certain plant materials or animal materials, and we are using and we are we want to dehydrate the material, another step is incorporated in this before staining, that is the dehydration step. There also we are using different grades of alcohol to dehydrate the material, then finally staining, and finally observation. Okay, so these are normal steps which we normally follow when we prepare samples with light microscope. But in EM or the electron microscope, mainly in case of TEM, a very tedious specimen preparation technique is required. So I am not going detail, but just mentioning the steps and what is the difference with the light microscope during the specimen preparation technique. Okay, so we have learned that in light microscope we do fixation mainly with heat. Okay, but in case of specimen preparation technique with EM, we use the chemical fixation technique. Okay, always. So never we do the heat fixation technique. We use the chemical fixation technique, which is mainly done with the aldehydes like acetaldehyde or glutaraldehyde, or sometimes osmium tetroxide fixation techniques is also used. And then is the dehydration steps. And here also, as in with plant material in case of light microscope or animal material with light microscope, we are using different concentrations of ethyl alcohol. Okay, so by just increasing the concentration of ethyl alcohol, we dehydrate the material. Then the specimen is embedded in epoxy resins to prepare a block. Okay, if sectioning is required in electron microscope, we cannot hold a bacteria, right, to cut sections. So we have to prepare certain blocks so that where the specimens are embedded, right? And after that, we just cut the sections. So I think you can you also having some experience who are having botany or zoology in your subjects. They in botany mainly. If we are trying to cut certain sections of certain thin leaves, we sometimes made a block of potato or carrot and make a slit over there and put the slender material in that slit, and after that just to make a block, right? So after that we cut sections to that block so that we get the sections of the thin leaf itself, right? So in that way, so that is the technique applied for section cutting in case of light microscope. But here the sections are the specimens are initially embedded in certain we can say wax-like material, not actually wax. Here the epoxy resins are used to prepare the block. Initially the wax were also used, but wax as having certain drawback, it was replaced with this epoxy resins. Okay, so initially the specimens are embedded in this epoxy resins and to make a block, and then sections are cut through using ultra microtomes. Okay, and specimens are taken in copper grids, right? So metallic stains, obviously, as in case of light microscope, we are using certain chemicals like you have already learned about the crystal violet, safranin, or there may be malachite green, like that. So here the metallic stains like uranyl acetate, lead citrate are sometimes used, and negative stains like phosphotungstic acid that also may be used. And sometimes they are coated with gold, and silver containing glue sometimes may be used. So these are some special techniques which we are applying in this in the specimen preparation for the electron microscopes. And here we are just you can see this is the copper grid, okay, and which is normally 3 millimeter in diameter, and this mesh size, the mesh frequency may be variable according to our need. Okay, so larger mesh or maybe very closely associated meshes may be used in this copper grid. And this is the microtome machine where we just cut the sections. And for electron microscope, we required the sections which are nearly 50 nanometer to 60 nanometer in thickness. Okay, so this type of section cutting, this is the resin block. And here one thing I have to mention, that the ultra microtome cut sections with diamond. Okay, so these are the thin sections which are floating in the water, and after that with the certain forceps or like that or brush, you just transfer it in the copper grid, and ultimately they are coated with some, they are fixed with some glues, and ultimately coated with some materials like gold or like that, and they are ultimately observed under microscope. So this is all about the microscope part, and what are the main differences in the, I'm not, again I am saying I'm not going to great detail of the steps of the specimen preparations, because there are lots of modifications according to your specimen. What type of fixative you have to apply, what type of stain you have to apply. There are lots of modifications and optimization according to the specimen you want to view under EM. But the basic difference between the between the sample preparation of light microscope and the sample preparation in electron microscope, I have discussed. Okay, so this is my mail ID. You can feel free to contact for future correspondence. Thank you.
Hello, ma'am.
Yeah.
So, is there any specification for using copper only as a grid, or can we use any other alloy or polymers?
Normally, copper is used, and I read in somewhere molybdenum is also used as grid material, but mostly used material is copper. Okay.
Yes. So, is it the conductivity of the metal?
Yeah. That is, I think that is more important over here.
So, if a sample itself has an conducting nature or some other element which is able to channel electrons.
I think this copper is optimized, right? So having less distortion or like that, okay, so that the copper has been selected. I told you now there are lots of optimization in each and every steps over there in the specimen. Okay, but universally copper grids are used. Okay.
And ma'am, with regarding to TEM imaging, do we get a proper 3D image or is it a flat image?
Normally, in case of TEM, it is 2D, but in 2D. Case of in case of SAME, it is 3D. Okay, and there are lots of techniques also. I have already mentioned the freeze fracturing and shadowing technique is very important. In that case, we get a much better 3D image.
Yeah, so I would like to request other participants to please come forward with their doubts.
Hello, ma'am.
Yeah.
Ma'am, I have one trivial question, by the way. I have always been wondering that whenever we see a specimen or visualize a specimen from through transmission electron microscopy, for any of the specimen there has to be a cell layer, I mean like a lipid layer and everything, a cell wall or a cell membrane has to be there. For visualizing the internal cell organelles, how do we prepare the cell? How do we visualize the internal organelles without removing the upper part? Like we have a skin, in order to visualize the underneath parts of the skin, we'll have to first peel out the skin and then see what is there.
Normally in case of TEM, there are sectioning is done. First sectioning is done and then you can visualize the whole thing. You have to make sections of the cells first, then you can visualize the internal part. There is obviously a surrounding membrane means not the surrounding membrane, the outer part of the surrounding membrane is visible. Otherwise, and the internal part is visible only after section cutting. So sectioning is very vital part in the sample preparation of TEM. Without cutting section, you cannot view anything, right? Yeah.
Okay. Ma'am, in this section cutting, if we suppose the problem that suppose when we do a freeze fracture, At that point of time, we have a specific procedure so that the specimen that we really want to visualize. So, for example, if you want to visualize the cell membrane, it should not be distorted. So whenever we study the hello. Sorry.
No, it's okay. Continue, press on.
Yes.
Okay, so whenever we visualize any of the structure through transmission electron microscope, whenever the steps are being described, we do not have very specific steps for how to prepare the specimen so that only internal structures are visualized and they are not distorted while preparing the specimen. So therefore, I was really wondering, because whenever we study SEM and TEM, the steps that are involved, they just describe that preparing the specimen in terms of what are the coating that is there, so that electrons are actually some are to be transmitted. But exactly just like in freeze fracture, whatever the steps that are involved so that the internal structures are intact and they are not distorted. In that particular way, I have never studied the internal like preparation of the specimen. Therefore,
see, I have not that much expertise to preparation in that part. That is the specimen preparation of that TEM. But I think there are certain methodologies are there just to not distort the internal parts and just to in the freeze fracture mainly the I think the knife is very and obviously the technician should have that level of expertise to cut the section so that the internal parts are not hampered. I think so. I don't have that much expertise. I have just done during my PhD work one time, but that was one technician was there, and he just prepared the material, then cut sections and do the staining whatever is needed, and ultimately we just found the image like that. So I think the expertise is available over there, and there are certain special techniques are there so that they can cut sections without hampering the internal structures or like that. Okay.
So thank you, Prasoon, for your question. So we have another question from Sedu Lakshmi. So she wants to know, for understanding material composition, which type of microscopy should we go for? Is it TEM or between TEM and SEM?
Material composition in which, what sense?
Sedu Lakshmi, could you please clarify?
Biological material. So see, if you are if you just want to have the outer structure, that is the superficial structure, the 3D imaging, you should use the same. Whereas the internal structures can be done with the TEM, right? So that is the main point. So material composition, I think for composition, microscopy is not that much important. You have to do other type of analysis like that. So like other methodologies are there, not only the microscopy only for just for imaging or like that. Okay, so for determining the material composition, you have to do either other type of work like XRD or like the what I can say, the different types of gel filtrations or identification like that. Okay, mass spectroscopy. So or NMR spectroscopy to know the composition of the material. So you can just have the image with this same or TEM. So either the superficial or the internal. So if you want to get the superficial structures, you should go for SEM, and for the internal structures you go for TEM. Right?
Sedu Lakshmi, I hope that clears your doubt. If any still persists, please drop it in. Okay, so anyone else who would like to ask something from Ma'am? Okay, so for follow-up questions, Ma'am has provided her email account.
ID. Yeah.
Yes, so please feel free to drop in her mail. So thank you so much, Ma'am. It was a wonderful talk. And we would again like to thank you for the insights about working of different types of microscopies which you have talked about. I'm pretty sure when we will have these points in mind from now on when we are analyzing our own samples under the microscope. So on behalf of PGAC team, we would like to present our token of gratitude for your time and effort, ma'am. And I have the honors to present it to you.
Thank you so much.
Rashmi, will you please share your screen? Rashmi, yeah.
Oh, thank you so much for the wonderful certificate. Thank you the whole team for arranging such a session and invite me to deliver my thoughts, my ideas with you. And thank you all the participants.
Thank you so much, ma'am. So I'd like to request Miss Rashmi to conclude the session with her vote of thanks.
I, on the behalf of all the participants of IIT Bombay, extend my hearty vote of thanks to Professor Mahuya Mukhopadhyay, ma'am. Thank you, ma'am, for accepting our invitation and delivering a wonderful talk on the fundamentals and applications of microscope. Thank you, ma'am. A very big thanks to all the participants for taking time and attending this webinar. Lastly, and most importantly, I would like to thank my PGAC team, Sohini, Preeta, Manu Krishnan, for being the backbone in conducting this 4-day webinar. That's it. I request all the participants to please fill in the survey form, which is provided in the link, which is provided in the chat box. It will be helpful for improving our further webinars. Ma'am will also be sharing her presentation with us. Thank you, ma'am. Thank you, ma'am.
Thank you so much.
I will end my
Thank you so
extending
much.
the thanks again. Thank you.
Thank you.
So yeah, I would request the participants to please help us with the survey forms. We will be following up with the certificates shortly. Thank you for your participation. Have a good day.
Thank you so much, and we will be mailing the certificate to ma'am in a short while.